Palatine chromosomes. Palatine chromosomes are also known as salivary gland chromosomes. These chromosomes were observed for the first time by E. G. Balbiani in 1881. He was able to see these chromosomes in Chironomus. Actually, Chironomus, mosquito, Drosophila, and houseflies, even several pest flies like melon fruit fly. In their larval stage, Polytene chromosomes can be seen, particularly in their salivary glands. So that is why these chromosomes are also referred as salivary gland chromosomes. Although these chromosomes can be prepared from several secretory glands like Malpighian tubules, midgut cells, adipocytes, certain cells of ovary. They are secretory in nature, and from such cells, the Polytene chromosomes can be prepared. But the best preparation of Polytene chromosomes are very good size of these chromosomes can be prepared only from the salivary glands. Now, these chromosomes are found to be in interphase cells, that is, they are found in the nuclei of interphase cells. Each Polytene chromosome is 200 to 600 micrometer long, and these are formed by endomitosis, that is, multiple rounds of DNA replication takes place, where there occurs duplication of chromatin content, but cytokinesis does not occur, and that is why the uh, chromosomes look quite large in size. Large means lengthwise such chromosomes are long because the chromosome contents are in interphase, and so many duplications of DNA or DNA copies will be there. Maternal paternal chromosomes, that is the two homologous chromosomes, they remain in homologous conditions, and they are tightly synapsed, tightly arranged uh, with each other, and that is why such chromosomes look very thick. Poly means several, many, teen means thread. So such chromosomes are made up of so many threads, and threads means the chromatids, maternal, paternal chromatids, they are arranged in parallel manner, and that too in thousands of copies. So that is why these chromosomes are large chromosomes. They are also referred as giant chromosomes. Now, if you prepare the metaphase chromosomes from the somatic cells like brain cells of a larva, you can see this kind of arrangement. This is actually metaphase chromosome karyotype, where you can see these two lower chromosomes are X chromosome. So two X chromosomes are arranged in case of female, and then in the left side you can see these two are suppose second chromosome. These are third chromosome in the right side, and the tiny dot-like structures are fourth chromosomes. So as a whole, if you count the number comes 8. So 2n is equal to 8. And exactly the same number will be there in male. Only thing is that there will be X and Y chromosome, because in Drosophila the sex determination is XY type. So two X chromosome, that is two sex chromosome, and then three pairs of autosomes are found in metaphase chromosomes. But the Palatine chromosomes, they look very differently. In case of Palatine chromosome, you will find that there is a dark area in the central portion that is referred as chromocenter. And from this chromocenter, 5 chromosome arms actually radiate around it. So what happens? The centromere of all these chromosomes actually fuse with each other and make this common area, and that is chromocenter. It is heterochromatic part of the Palatine chromosome. And why we observe 5 chromosome arms? Actually, the maternal and paternal chromosome, like these two chromosomes, they will be completely synapsed throughout the length. Likewise, the second and third chromosomes, they are also permanently synapsed all along their length. And the centromere portion that gets fused here, so both arms will be radiating. but the X chromosome will be a single arm. So here the X chromosome is visible. There is a single, arm of X chromosome in Drosophila melanogaster, and then you can see 3L. 3L means left arm of third chromosome. Then you can see 2L, that is left arm of second chromosome. This is 2R, that is right arm of second chromosome, and this one is 3R, that is the right arm of third chromosome. So 3L, 3R are left and right arm of third chromosome, 2L, 2R are left and right arm of second chromosome. And the X chromosome being acrocentric or almost allocentric, that is represented in the form of a single chromosome arm. Had this particular species possessed, actually this is the structure of Polytene Chromosome of Drosophila melanogaster, in which the X chromosome is acrocentric in nature. But in certain species, you would be able to see two X chromosome arms in case if it is metacentric or submetacentric chromosome. In that case, you would be able to see six chromosome arms instead of five, because the X chromosome will also be seen as XL and XR, and that can be seen in case of Drosophila ananaceae. Other important point is that by seeing the banding patterns, see Palatine chromosomes, they have bands and interbands. In certain area, you can see the dark band. These are the highly condensed portions. And then you can see the light portions. These portions are euchromatic portion where the condensation is not of that type. So that is why you will find that light and dark regions are there. And based on the binding pattern, one can identify the individual chromosome arms. So this is beauty of such chromosomes, that they are quite large in size. They are also referred as giant chromosome, and because of definite patterns of bands and interbands, one can observe all sorts of structural chromosomal aberrations like deletion or duplication or inversion, even translocations. These four major structural chromosomal aberrations can be seen in the Palatine chromosome. And if one chromosome arm, that is paternal one, is a normal one, and the maternal possesses deletion or duplication or inversion, in that case, you would be able to see a heterozygous loop. So the presence of a heterozygous loop indicates that that particular area possesses some kind of structural aberration, and that too of heterozygous nature. So by seeing the banding pattern, one can know that what kind of aberration exists over there. Inversions are very common in Palatine chromosome, particularly paracentric inversions. That is those inversions which do not include centromere in their limit. So in case of Drosophila, the paracentric inversions can be observed, and some of them are cosmopolitan, or they are ubiquitous, means worldwide in distribution. Since they are selected, they provide some fitness to the individuals who carry them, so they are retained in the population to a very large extent. You can see the presence of puffs. Certain areas will be there where the chromosome portion will be very loose and will be a light area. So that area is that area where RNA synthesis or transcription takes place to a very good extent. So the transcription aspect can be studied by using Palatine chromosome, because chromosome puffs, which are known in the form of Balbiani ring in Chironomus, are the areas where active transcription or active synthesis of RNA takes place. Then some of the important use and function of this Palatine chromosome could be that in Palatine chromosomes, multiple copies of genes are available due to endoreplication that provide a high level of gene expression. Then chromosome puffs are the sites of active transcription, that is synthesis of RNA, which gets synthesized ultimately into specific proteins. Appearance of puff at varying regions of Palatine chromosome arms with varying larval stages indicate differential expression of genes with age. And finally, the cells with Palatine chromosomes are involved in intense secretory functions. for rapid growth and differentiation of the individual. So these are the major salient characteristics of Palatine chromosomes. Thank you.